In this lesson, we'll be looking at the importance of microscopes to biology and at some of the issues involved with using enlarged images. The specification has the following to say about this. Students, the size of the real object. In the 1st century AD, Roman philosophers noticed that they were able to see an enlarged image of objects when they viewed them through a clear spherical flask filled with water, or a piece of clear glass that was thicker in the middle than at the edges. The images were very unclear though, and they didn't increase our knowledge of the natural world. As the magnifying power of a curved, clear surface became more widely known, magnifying devices were developed. From the 8th century, spherical segments of clear minerals were used as reading stones, but these were mainly used to help older people to see better. In the 13th century, clear silicate glass was discovered, and this allowed proper spectacles to be developed. The low magnification and the poor image quality of these early devices did little to advance our understanding of living things, though. Now, early in the 17th century, the Englishman Robert Hooke was the first person to use a microscope with 3 lenses, and that's the configuration that is used in most of today's modern microscopes. He used this microscope to make elaborate drawings of living things. His drawings showed far more detail than had ever been seen before by other people. Hooke was the first to observe cells, and he gave them that name. In the late 17th century, Anton von Leeuwenhoek from Holland invented a single-lens, handheld microscope that could achieve an even higher magnification of around 270 times. With his microscope, he was able to see structures that no one had ever seen before, such as blood cells, and even new organisms, such as bacteria and protozoa. Hooke had examined the dead tissues he found in cork. In the following century, the German botanist Matthias Schleiden studied living plant cells. He saw that their contents moved within and between the cells and along fibers composed of elongated cells joined end to end. Schleiden called this process protoplasmic streaming. The protoplasm outside the cell nucleus that he saw is now known as cytoplasm. Schleiden concluded that all plants are made up of cells. The following year, Theodor Schwann came to the same conclusion about animal cells. Most of the significant advances in our knowledge of cells have depended on the resolving power of the instruments we use for magnifying these tiny objects. The resolving power of an instrument is the distance separating 2 objects that allows them to be seen as 2 distinct things rather than a single object. For example, most human eyes can see 2 fine parallel lines as distinct markings if they are separated by at least 0.1 millimeter. But if they're any closer than that, we see them as a single line. So the resolving power of a typical human eye is around 0.1 millimeters, which is about the same as a human egg cell. Now, the development of the light microscope made the study of cells possible. The resolving power of a microscope is about 0.0002 millimeters or 200 nanometers. So it reveals some subcellular organelles. This resolving power cannot be improved by adding more lenses though. An enlarged image retains the same resolution and only becomes fuzzy as the image gets bigger. No more detail is added. In the last half-century, the invention of the electron microscope showed many structures that are far too small to be resolved with the light microscope. In essence, the unaided eye can see multicellular structures. The light microscope can see individual cells and some of the larger organelles, but the electron microscope can see details of the individual structures within the cell. Ribosomes, for example, being about 20 nanometers or less in diameter, can't be seen using a light microscope. An electron microscope uses powerful magnets to focus an electron beam, in a similar way that light microscopes use glass lenses. Electron beams have a much shorter wavelength than beams of light, so the probing beam can fit between spaces that a light beam wouldn't be able to, and that leads to a far better resolving power. We can't see electrons though, so they are directed to a fluorescent screen to create an image that we can see. The resolving power of an electron microscope is theoretically around 0.2 nanometers. But that's difficult to achieve with biological specimens because the energy of the electron beam at that power is so great that it destroys biological molecules. In practice, the usable resolving power of an electron microscope is around 2 nanometers. That's around 100,000 times better than the human eye. There are two types of electron microscope. In transmission electron microscopes, electrons pass through the sample. These are used to examine very thin slices of objects, similar to the sections that are shaved off a material onto the slides of a light microscope, but much thinner. In scanning electron microscopes, the electrons are bounced off the surface, and that gives a three-dimensional aspect to the image. Scanning electron microscopes reveal surface details of three-dimensional objects. The resolving power of these microscopes is around 10 nanometers, so the images that are obtained tend to be of lower magnification than those from transmission electron microscopes. Despite their high resolving power, electron microscopes are not suitable for all microscopic studies. Living specimens can't be used. The apparatus is very large and it's very expensive, and it requires lots of training to be able to use it properly. Now, in a modern light microscope, there are normally two places where you need to take into account the magnification which takes place: the eyepiece lens and the objective lens. The eyepiece lens is normally times 10, and the objective lens has a range of different fixed values: times 4, times 10, times 40, times 100. The overall magnification is found by multiplying the magnification at the eyepiece and the objective. To judge the size of an object viewed under the microscope, you need to use the following formula. The actual size of an object is equal to the size of the image divided by the amount of magnification used. Now, manipulating algebraic expressions like this in an exam can often go wrong, even for the most naturally gifted mathematicians. However, examiners can still give close to full marks if you show your workings and they can see which parts were done correctly. But if you just write down the final answer and it's based on an error, you won't get any marks. So for this reason, I recommend getting into the habit of always clearly setting out all the steps in your answers, even the blindingly obvious. So using the formula triangle to show the expressions that you use. In this type of question, you'll usually need to measure part of an image, but there might also be other pieces of vital information on the diagram. such as a scale bar or a note of the magnification. Just make it clear in your working as you use each piece of information. Okay, it's question 1. Find the size of this mitochondria in micrometers. First step is to set out the magnification equation. Next, the magnification equation has 3 parts to it. We can get 2 parts of this from the picture, and that allows us to calculate the missing 3rd part. Next, we need to accurately measure the image size. Next, we place the figures we have into the equation that we set down earlier. And we need to convert this to the units that were asked for. Now, if you're not sure about this, you need to see the lesson on math skills, part 1. Question 2. Find the magnification of this image and use it to find the size of the white blood cell in millimeters, expressed in standard form. As before, the first step is to set out the magnification triangle and take notice of which pieces of information we've been given. At first glance, this seems a bit more tricky because we're missing 2 of the 3 parts of the equation, not just 1. However, the scale bar has actual size on it, and we can measure its image length. And that gives us the magnification being used. With the magnification used for the cell image and a measurement of its size, we can use the magnification equation to calculate the actual size of the cell. Don't forget to download the resources for this video from the link in the description, especially the notes template. LearnFix has a lot more to offer than just the videos and the exam questions. The most popular aspect is the set of 12 online learning games we have for each topic. These help you practice your recall of the details. Now, when it comes to revision, LearnFix is there to help. After each section of the syllabus, there's a diagnostic quiz to help focus your revision efforts. You can then use a progress report to look back at the details for that particular topic, what went well and what needs more work. We're adding new features every month, so visit us at learnfix.com to find out more.